Can we live forever? What if you never aged? What if the relentless march of time simply stopped affecting your body? Imagine your reflection in the mirror remaining unchanged for decades, even centuries. Picture your mind, sharp and clear, accumulating knowledge and experience far beyond a normal human lifespan. These are profound questions, the kind that echo through human history in myths and legends. But today they have moved from fantasy into the cold, hard light of the laboratory. We are no longer just dreaming. We are investigating. We are asking, is biological immortality truly possible for a human being? Would your body actually withstand the test of 1,000 years? If you appreciate questions that challenge the very definition of being human, and you want answers grounded in evidence, I invite you to subscribe to this channel right now. We dig deep into the facts. We are here to explore the universe, and that includes the universe within you. The journey begins with a single, powerful question. We will not shy away from the complexity. We will confront the data head-on. This isn't about wishful thinking or science fiction. It is about the real, tangible science happening in labs around the world as we speak, on this day, 2025, December 23rd. Let us be absolutely clear about our mission here. We are going to look at the hard science of aging. We will examine the peer-reviewed studies, the clinical trials, and the biological data that form the foundation of this research. We will leave vague philosophical debates for another time. Our focus is squarely on biology, medicine, and the physical realities of the human body. I am speaking directly to you because this conversation is about your cells, your DNA, and the future of your own life. It is one of the most personal scientific inquiries you can undertake. The quest for immortality is not a simple yes or no question. It is a maze of biological pathways, genetic codes, and ethical dilemmas. To navigate this maze, we must start at the beginning. We must understand the fundamental enemy we are trying to defeat, the process of aging itself. Why do you get wrinkles? Why do your joints ache? Why does your body eventually fail? The answers lie deep within your cells, written in a biological language that scientists are only now beginning to translate with any real fluency. So let's get started. Section 2, the machinery of decay. Why your body ages. To understand how we might stop aging, you first must understand why it happens at all. Aging is not a single event. It is a slow, cascading failure of multiple systems within your body, starting at the microscopic level. Think of your body as an incredibly complex machine. Over time, its parts begin to wear down. Cells, the fundamental building blocks of this machine, accumulate damage. They become less efficient. They slow down their functions. They eventually stop working as they should. Scientists have identified several key culprits behind this gradual decay. One of the primary drivers is the accumulation of DNA damage. Your DNA is the blueprint for everything your cells do. Every single day, this blueprint is under assault. Ultraviolet radiation from the sun, chemical toxins from the environment, damaged by the byproducts of your own metabolism. Your cells have sophisticated repair mechanisms that work around the clock to fix this damage. However, these repairs are not always perfect. Over many decades, small, uncorrected errors and mutations build up, leading to cellular dysfunction, disease, and the visible signs of aging. Another major factor is mitochondrial decline. You have probably heard mitochondria called the powerhouses of the cell, and for good reason. They convert the food you eat into the chemical energy that fuels every single biological process. As you age, your mitochondria become less efficient and produce more harmful waste products, known as free radicals. This decline in energy production means your cells and organs have less power to perform their functions and to repair themselves. It is like trying to run a bustling city on a failing power grid. Eventually, systems begin to shut down one by one. Finally, there is the problem of cellular senescence. This is a state where cells stop dividing but refuse to die. they become zombie cells. These senescent cells are not just inert, they actively secrete a cocktail of inflammatory molecules that harm neighboring healthy cells. This creates a low-grade chronic inflammation throughout your body, which is now recognized as a major contributor to many age-related diseases, arthritis, heart disease. As David Sinclair, a prominent Harvard biologist and longevity researcher, has stated, aging is a disease, and that disease is treatable. This perspective frames aging not as an inevitability, but as a biological problem with a potential solution. Section 3. The Hayflick limit. Your cells have a clock. In the 1960s, Leonard Hayflick, Paul Moorhead. They discovered that normal human cells in a petri dish will only divide a finite number of times before they stop. The Hayflick limit, typically around 50 to 70 divisions. Once cells hit this limit, they enter senescence, the zombie-like state. The physical basis lies at the ends of your chromosomes, telomeres. While humans and most other animals are bound by aging, the natural world is full of exceptions. By studying negligible senescence, a lack of age-related decline, we may uncover tricks for human health. These creatures show aging isn't universal. They can slow or reverse the biological clock. They offer a blueprint of what's possible. Immortal jellyfish, Turritopsis dohrnii. When injured, stressed, or starving, it can revert adult cells to juvenile polyps. It hits the reset button on its life cycle. From the polyp, it regrows into a new adult. It can still die from predators or disease, but it escapes death by old age. This is cellular reprogramming in action. Lobster. Lobsters produce high levels of telomerase in many tissues. Telomerase rebuilds telomeres and counteracts shortening. They avoid a typical Hayflick limit. Lobsters keep growing larger, stronger, and more fertile with age. They usually die from molting disease or being caught. Naked mole rat. These rodents can live over 30 years. A common mouse lives 2 to 3 years. Naked mole rats resist cancer and stay physiologically robust. They clear damaged proteins better and have a highly stable genome. Their biology gives direct clues for mammalian aging research. The average human lifespan has increased dramatically over the last century, from around 31 years in 1900 to over 73 years globally today. This is primarily due to advances in sanitation, nutrition, and medicine that combat external causes of death like infectious diseases. However, the maximum recorded human lifespan has remained stubbornly stuck at around 122 years. The Hayflick limit and the telomere shortening that drives it are considered one of the core biological barriers preventing us from living much beyond this point. Section 5. The Scientist's Toolkit. Hacking the Code of Life. Armed with the knowledge of why we age and the examples from nature's outliers, scientists are now developing a powerful toolkit to intervene in the aging process directly. This is no longer theoretical science. It is an active field of biomedical engineering. Researchers are not just aiming to treat the diseases of old age, like heart disease or Alzheimer's. They aim to treat aging itself, the root cause of these conditions. The strategies are diverse, targeting different aspects of the aging machinery, from our genes to our cells. One of the most powerful and promising tools is gene therapy. This technology allows scientists to directly edit the source code of life, our DNA. Using tools like CRISPR-Cas9, it is theoretically possible to correct genetic mutations that accelerate aging or insert genes that confer longevity. For example, scientists could introduce genes borrowed from long-lived species or versions of human genes associated with exceptional health in old age. While still early for complex conditions like aging, gene therapy has already been used to treat several rare genetic diseases, proving the principle is sound. Another major area of focus is the development of senolytic drugs. Senescent zombie cells accumulate with age and secrete harmful inflammatory signals. Senolytics are drugs designed to selectively identify and destroy those cells, clearing them from the body. In animal studies, clearing senescent cells has been shown to rejuvenate tissues, improve organ function, and extend healthy lifespan. Several senolytic drugs are now in early-stage human clinical trials. Artificial intelligence is revolutionizing the field. Biology's complexity is a massive challenge. AI and machine learning can analyze vast data sets, genomic cellular behavior, to identify patterns that a human researcher might miss. AI can predict how a novel drug molecule will interact with proteins, speeding drug discovery. It can identify new biological targets and personalize treatments based on an individual's genetic makeup. This computational power is an essential accelerator in the race to understand and control aging. If this is blowing your mind, like the video, share it, and comment whether you'd choose to live forever. Even if we could perfect the biological tools to halt or reverse cellular aging, achieving true immortality would confront us with other, more formidable hurdles. A longer life is a longer time for things to go wrong. The two most significant biological barriers that stand in our way are the ever-present threat of cancer and the unknown limits of the human brain. These are not simple engineering problems. They are fundamental aspects of our biology that may prove incredibly difficult to overcome, even with advanced technology. The link between aging and cancer is undeniable. Cancer is, at its core, a disease of accumulated genetic mutations. The longer you live, the more cell divisions your body undergoes, and the more opportunities there are for a random mutation to occur in a critical gene that controls cell growth. It is a game of probability. Living for 150 or 200 years would dramatically increase the statistical likelihood of developing multiple, independent cancers over your lifetime. Many of the proposed anti-aging interventions, like activating telomerase, could inadvertently increase this risk. An immortal body would require an immortal system of cancer surveillance and destruction, something far beyond our current capabilities. Then there is the matter of your mind. Your brain is the seat of your consciousness, your memories, your very self. It is an organ of staggering complexity, composed of 86 billion neurons forming trillions of connections. We simply do not know if this biological hardware is designed to function for centuries. Let us engage in a thought experiment. Imagine that tomorrow, a safe and effective cure for aging becomes widely available. What would our world look like? The societal and ethical implications are just as challenging as the biological ones, and they must be considered. Immortality would not just change individual lives, it would fundamentally break the systems our civilization is built upon. Social systems, economic systems, demographic systems. The cycle of generations, of birth, life, death, is the engine of social change, and we would be switching it off. The most immediate problem would be population. If people stopped dying of old age, and death only came from accidents or violence, the global population could explode. This would place an unimaginable strain on our planet's finite resources: food, water, energy, living space. Who would get access to these resources? Who would get to have children in a world where no one makes way for the next generation? These questions could lead to unprecedented global conflict and inequality. The world would have to implement strict population controls, raising profound ethical questions about reproductive freedom. If this topic is sparking your curiosity, I urge you to like this essay and share your thoughts in the comments. Section 6. Telomerase, the double-edged sword of cellular youth. At the heart of the cellular clock is the enzyme we mentioned earlier, telomerase. This remarkable molecule adds DNA back onto the ends of chromosomes, rebuilding telomeres lost during cell division. In most of our body's cells, the telomerase gene is switched off shortly after birth. This is why our telomeres shorten over time. However, some cells keep telomerase active. Reproductive cells, sperm, egg, some stem cells. These cells must divide many times without aging to create new organisms and replenish tissues. The potential of telomerase as an anti-aging therapy is obvious and profound. If we could safely reactivate telomerase in our aging cells, we could theoretically rewind the Hayflick clock. We could lengthen our telomeres, allowing our cells to divide and rejuvenate tissues for much longer. In 2012, a landmark study in mice led by Maria Blasco showed just this. Genetically engineered mice that produced extra telomerase lived up to 24% longer than normal mice, and they were healthier. Lower incidence of age-related diseases, and better metabolic function. This was a stunning proof of concept. However, telomerase is a classic double-edged sword. There is a very good reason that nature switches it off in most of our cells. The ability for a cell to divide indefinitely is not just a hallmark of youth, it is also a hallmark of cancer. In fact, around 90% of all human cancers have found a way to reactivate telomerase. That lets them bypass the normal cellular aging process and achieve a dangerous form of biological immortality, dividing uncontrollably to form tumors. Activating telomerase throughout the human body could be like pouring gasoline on the smoldering embers of precancerous cells that we all carry. Some proposed strategies involve transient activation of telomerase, switching it on briefly to lengthen telomeres, then switching it off again, or combining telomerase therapy with powerful cancer surveillance techniques. Until this safety issue is definitively solved, widespread use of telomerase activation in humans remains a high-stakes gamble that few are willing to take. Imagine a garden where old, withered plants are not removed. They remain, taking up space and releasing substances that harm healthy plants. Over time, the whole garden becomes sick and unproductive. This is an analogy for cellular senescence in your body. As you age, non-dividing zombie cells accumulate in tissues. These cells actively secrete a damaging mix of inflammatory proteins, the senescence-associated secretory phenotype, or SASP. The SASP poisons the local environment and accelerates aging of neighboring cells. This is where senolytic therapies come in. Senolytic literally means destroyer of the old. Senolytics are drugs or compounds that trigger apoptosis, programmed cell death, in target cells. They selectively induce death in senescent cells. Healthy cells are left unharmed. The strategy isn't to stop senescence. It's an important anti-cancer mechanism. Instead, the idea is periodic clearance, a cleanup crew removing cellular trash. In a Mayo Clinic study, researchers cleared senescent cells periodically from mice. The treated mice lived longer. They had healthier hearts. They had healthier kidneys. They were more physically active. They were less likely to develop cataracts. They were less likely to develop other age-related conditions. The intervention extended health span as well as lifespan. Dozens of follow-up studies across models have replicated and expanded these findings. That makes senolytics one of the most tangible anti-aging strategies today. But will it work in humans? Several human clinical trials are underway, testing senolytics for osteoarthritis, for idiopathic pulmonary fibrosis, and for chronic kidney disease. Initial results are cautiously optimistic. Lower senescent cell burden and reduced inflammation. Still, these are early days. We need larger, longer trials to confirm safety and systemic effectiveness. We began with a series of profound questions about the possibility of eternal life. We have journeyed through the cellular mechanisms of decay, shortening telomeres, senescent zombie cells. We have looked to the animal kingdom for inspiration, gene therapy, senolytics. We have also confronted the formidable hurdles of cancer, brain limits, and massive societal disruption. After weighing the evidence, can science make you immortal? The honest answer today is no. True biological immortality endless life free from decay, is not on the horizon. The biological complexity is immense, the trade-offs are dangerous, and the systemic challenges are great. Every intervention, telomerase activation, senolytics, carries risks and only fixes part of aging's puzzle. Biology has fail-safes and unknown limits. Bypassing them could cause catastrophic unintended consequences, especially higher cancer risk. This is not defeat. It's realism and real hope. True immortality may be fantasy, but radical extension of health span is becoming possible. The goal: not immortal beings, but longer, healthier, functional lives free from debilitating disease. Science may extend healthy lifespan to 100 years, 120 years, 150 years. Optimists. Conclusion: humans will likely extend life, but not live forever. Living forever sounds simple, but biology makes it complicated. Subscribe to Doubts Undertaker, like this video, share it with someone who loves science, and comment what question we should bury next.